Okay, so we're transitioning to chapter 33 on bioenergetics. And it's mostly the parts in this chapter, you may have a test question or two directly on the parts to it, but it's more the application of what we learn in this chapter to 34 and 35 that'll be showing up on the test. And so everything in chapter 33 is certainly important. You have to understand it and able to do this stuff, but the test questions themselves tend to come mostly from chapter 34 and 35, if I remember right. So let's define bioenergetics. It's the transformation, distribution, and utilization of energy. Now, that would be a great definition for even chemistry for the study of energy. Of course, if we add the word bio in it, we have to say by living organisms. All biology really is simply chemistry with the word "and for living organisms" thrown on top of it. That's not totally true, but it's somewhat true. So if we take a look at this, what we're going to do is we're going to look at how it's transformed and how it's sort of distributed, how we can follow it around through various processes in the body in chapter 33, and then the utilization, the specific biological cycles that we're going to look at, things like the Embden-Meyerhof pathway. We'll probably skip the citric acid cycle since that's the one you do in Todd's class, right? Am I right? And so we'll skip that one usually. We usually do beta oxidation, and then we start talking about proteins. Proteins can be used for energy. They're usually used for other purposes, mostly to supply the amino acids we need to build proteins. And so when we get to that chapter, we usually focus on that a little bit more. And so this is what we're going to learn in chapter 33 and chapter 34. And like I said, we're learning all the tools here. We're going to focus on the specifics, things like the Embden-Meyerhof pathway. We're going to look at beta oxidation of fatty acids. This is for glucose or carbohydrates if we want to be very generic. And then we'll also look at lipogenesis. And those are for sure the three that we always hit. And then depending upon time and how things go, we may hit more or less of some of the other topics. But that's kind of the main focus that we're going to be doing. So metabolism then, which is basically the sum of all the chemical reactions, is the sum of all chemical reactions. You guys know the sum symbol? This means just sum. It's from math. In a living organism. And this is basics for biology. I'll bet you Todd's covered some of this. Maybe Sarah's covered some of this. Certainly if you've taken Bio 111 and Bio 112, you've kind of talked about it. What two categories can we break down metabolism into? We can talk about anabolism. Maybe we don't need numbers there, I don't know. And catabolism. Does anyone remember what the difference between those are? So it breaks up. So it builds molecules up and breaks molecules down. This is kind of like what's the words I want? Oxidation and reduction, meaning they're kind of paired. You can almost always have an anabolic process. You'll also usually have a corresponding catabolic process with it and things like that. So we build molecules up, we break molecules down. Some other things that we can kind of put to that are generally when we build molecules up, we reduce carbon. And the opposite for anabolism, if we are breaking molecules down, then that usually means we're oxidizing the carbon. And we will go and we will review oxidation reduction kind of shortly, just so that it's kind of clear in your head what we mean by those. Generally, in an anabolic process, the body uses energy. So we can say uses, or it sort of loses energy because it stores it in the molecule. And here, we usually release energy from the molecule. Release energy from the molecule. And actually, maybe I'll rephrase that one. Instead of talking about the body, we'll simply talk about the molecule. So here we usually store energy in the molecule, meaning the body has to use energy and there's a net loss for it. Some examples of anabolic processes that we'll look at is gluconeogenesis, close enough. We'll look at lipogenesis. What do you notice about both those words? Yeah, and genesis means to make things, right? And so, or at least that's, the world was made in Genesis in the Bible. And so glucogenesis is in chapter 34, lipogenesis is in chapter 35, and we'll also take a look, very, very brief look at photosynthesis in chapter 33. And for catabolism, what we'll basically look for or look at is the Krebs cycle, chapter 34, and we'll look at beta oxidation in chapter 35. So again, a lot of what we're talking about in chapter 33 is just parts and pieces that we'll use in chapter 34 and chapter 35 when we're talking about stuff. So this is kind of what I consider like a build-up chapter. Let's see. What do we do next? So your book has a lot of interesting energy level diagrams that it likes to draw. And so I know, we need to talk, going out of order here. We should talk about oxidation and reduction first. Oxidation and reduction. And you guys should hopefully be familiar with this by now. And so I'm going to have you guys fill in all the details. What would be a good definition of oxidation and reduction? That's one of the things. I'm not going to put that on top because that's not always a 100% sign, meaning generally if in a reaction you gain bonds to oxygen, you are oxidizing that molecule. However, the true definition is something more fundamental than that. And of course, for reduction, that means you lose bonds to oxygen. What would be another sign and or the fundamental part that we're looking for, that key? Yeah, the electrons is the real part of the definition. So if we lose electrons and if we gain electrons, because electrons are sort of the fundamental unit of energy that is being passed back and forth. Now your body converts that usually to ATP and to other forms, but from a chemist's perspective, it really is the electrons that are the true part that we're following. What about hydrogen? Is this kind of ringing a bell? We had it when we were talking about the energy difference between carbohydrates and fats. Didn't show up on that test. Guarantee you it will be on this test in some form or another. So in oxidation, the molecule loses energy. And so that means it shows up as a product in the reaction. And in a reduction, the molecule gains energy. Therefore, it shows up usually on the reactant side. And sometimes it literally will be energy. Sometimes it will be what I call energy equivalents, things like ATP and high energy phosphate bonds and substrate level phosphorylation and a bunch of stuff we'll learn after this.
So on the last homework for last chapter, when it said, or I don't know if it was homework, but it said like why if it is reduced it has more energy just because it gains it and it's a reactant, or
Well, we're storing electrons, and the bond between a carbon and a hydrogen stores more energy than the bond between a carbon and an oxygen. So every time we take a carbon-hydrogen bond and convert it to a carbon-oxygen bond, we get energy over here. Or conversely, if I do that reaction in the reverse order, every time I take a carbon-oxygen bond and reduce it to a carbon-hydrogen bond, then I'm storing energy in that bond. And so really this reaction goes both ways, right? In this direction, I'm doing an oxidation, and if I write it in that direction, or if I reverse the way I wrote it, I'd be writing a reduction. And that's one of those, I mean, you can actually go down to the fundamental level of following the electrons around too, but that's kind of like just a bare fact that you have to remember, that a carbon-hydrogen bond stores more energy in it than a carbon-oxygen bond. And the way I always remember it is the molecule that stores the most energy is methane, and the molecule that stores the least amount of energy is carbon dioxide. And notice that carbon dioxide has lots of bonds to oxygen, right? So this is what I would say is low energy. This is high energy. And then do you guys remember where we should put carbohydrates and fats in this? Which one contains more energy, fats or carbohydrates? Fats. And so if we take a look at a fat, you know, we'd put it there, and if I take a look at a carbohydrate, maybe I'll draw the carbohydrate this way. 3,4-CH2OH. We'll just draw all the OHs on one side because we don't care what one we're talking about, and then an aldehyde on the end. And so when we take a look at a typical atom, meaning the majority of the atoms in a fat look like a carbon with two hydrogens attached, and the typical carbon atom in a glucose molecule or a sugar looks like a carbon with a hydrogen and then an OH attached. And also there's two carbons on either side, but they're sort of energy neutral, meaning a carbon-carbon bond doesn't store or gain energy, at least usable amounts of energy. I should point out that anytime you make or break a bond, there's energy changes occurring. The question is whether we can extract any usable energy for it. So to me, that's how I kind of remember this. I usually just remember the extremes, and then if nothing else, I can usually remember that fats have more energy than carbohydrates. And then do you guys remember how we measured this? So we had some rules for calculating oxidation numbers. And oxidation numbers were one way to measure changes in oxidation and reduction. If we can calculate and assign a number to a carbon, we can state whether it's more reduced or more oxidized. Now again, 9 times out of 10 you can simply say I've gained or lost bonds to hydrogen and calculate it that and see it that way visually. Calculating it as if it's not obvious what happens. Or some people in the past have liked to calculate the numbers simply because it's a nice mathematical way to assign something, and once they assign it, you know the answer is right, assuming you didn't, add and subtract wrong. So you guys remember the basic rules for calculating those oxidation numbers? So hydrogen is always +1 in this class, and oxygen is always -2 in this class. Now, there are exceptions. For instance, if you talk about hydrogen peroxide, that's HOOH. In that case, oxygen is not a -2 in that compound. It's only a -1. And if you look at hydrogen bonded to metal atoms, then you have what are called metal hydrides. And again, in those cases, hydrogen is usually actually a -1 charge. And so there are exceptions to these rules, but when we're looking at living organisms, we don't worry about hydrogen peroxide because it's the exception to the rule. And I don't know any normal biological entity that uses metal hydrides somewhere in them. I mean, I'm sure you can find some weird exception that lives in like deep sea ocean vents or deep underground or something like that that has that possible, but I doubt it. So the other rule is that the sum of the oxidation numbers has to equal 0 or the charge. So if something is charged, then the sum of the oxidation numbers should equal that charge. And since we're dealing with biological molecules, 9 times out of 10 when we kind of look at them, we can simply say that the sum is going to be 0. And that's because we know that the opposites attract, positives and negatives, and that they, in a neutral molecule or in a molecule that's stable, you have to have equal amounts of both. So if I take a look at this carbon here, each of the hydrogens is +1, so what does the carbon have to be? Yeah, there's like 4 hydrogens times a +1 plus 1 carbon times, x equals 0, so it must be a -4. You can do the same thing on the opposite end. Oxygens are -2 and -2, so 2 times -2, that's for the oxygen, plus 1 times x equals 0. Well, x has to equal +4 in this case. And so one of the things that's unique about carbon, and I shouldn't say it's unique to carbon, but one of the most important parts about carbon and why it's kind of the central molecule of life, why it makes up most of the organic molecules that we're talking about, is because it can have an oxidation state ranging from -4 to +4. Hydrogen's always -1, hydrogen's always +1, oxygen is always -2. Did I say that right? I got the plus in this time. And so those don't have much flexibility, but carbon has a lot of flexibility because it can have that varying oxidation number. The other reason carbon's more useful is because it makes 4 bonds versus, say, nitrogen that only makes 3 and has a lone pair of electrons and stuff. And so that's another reason. And I'd say probably the last reason carbon's really important as a biological molecule is that it doesn't have any d orbitals to make chemistry complicated. So it follows pretty simple rules. And so, there's not a whole lot of weird stuff that happens with it. What about the carbon atom in a fat? If these are +1s, then what does carbon have to be? Does everyone see that? And then for the carbohydrate, it's a little harder because there's a +1, a +1. Oops, that's not 2 pluses. +1, +1, -2. What does carbon end up being? 0. And we could, of course, throw other examples in there like carboxylic acids, aldehydes, ketones, lots of other things that you could calculate the oxidation number for, right? So don't just memorize these 4 examples. Know how to calculate it for any possible molecule that you're given. Although if you think about it, you only have so many options, right? Meaning if you're looking at how many different combinations of carbon, oxygen, and hydrogen bonds you have, there's only a limited number. So you could try to learn them all, you could write them all down, but I think all in all, really, you're kind of mostly looking at, what's the word I want to use? Just kind of remembering the 3 rules, and it's a lot easier. It's like saying, you can memorize the multiplication tables for every number under the sun, or you could just kind of memorize them 1 through 10, and then everything else follows after that if you know the rule for how to carry and do things like that. Or if you just have a calculator. I don't know. Calculator. So what we want to be able to do is follow the energy in chemical reactions. Or I should say biological reactions, since this is biochemistry. And so one of my favorite things is they usually have these figures, and I kind of like them because they are a nice visual representation for stuff. But the general idea is that you should be able to convert the concepts into a diagram like this for any of the processes that we talk about. And so what they generally do is they place energy on one axis, and then they don't really have a second axis, but really there is. This is what we would call the reaction progress. And so, for instance, if we take a look at sort of the general cycle of photosynthesis to using things from our body, what we find is that we have energy being input. So this is h-nu. Does anyone remember what h-nu stood for? We've used it way back, like one of the first couple chapters in chapter 1. We've used it a little bit for other things. So this is really just saying light. H nu is the wavelength and frequency, or H is Planck's constant. Nu is the frequency of light. It basically is telling you the energy of light. And we use that squiggly line because light is, of course, a wave. But what else is light? It can be a particle too, right? So yeah, we got that whole wave-particle duality thing that, you know, we won't remember from last year. And so we have over here water plus carbon dioxide. And notice that these are low energy. Notice that specifically the carbon dioxide, you know, that we said is our low energy molecule, that has a -4 on that, +4 on that carbon. Oops. Kind of ran out of room, so we'll just draw an arrow like this, +4. And of course, we know that if we have photosynthesis, we make higher energy products. So this is our process, photosynthesis. And so we make reduced carbons, we make carbohydrates. So if we want, we can sketch our little carbohydrate. What's wrong with this picture? 1, 2, 3, 4. Oop, 5. Actually, I got it right. I thought I had an extra one there for a second. Plus O2 in our products. And of course, this is higher energy, right? Meaning one of the things the energy level diagram shows you is the energy levels. If we take a look at our average carbon here, we said that that was, let's see, +2, -2, 0 for our oxidation number. And so here, this is an energy storage process. Would we call this anabolic or catabolic? Yeah, this is an anabolic process, so we can kind of use that as a descriptor, anabolic. And are we reducing or oxidizing carbon here? Reduce. And so those are the type of questions that you're going to have to be able to follow. For the overall large processes like photosynthesis, and if we talk about the overall process in beta oxidation, you can know the answers ahead of time, meaning you can memorize things as being anabolic, you can memorize that something gets reduced. You can try to do that. The trouble is by the time you get to the test, you're going to have photosynthesis, beta oxidation, which is like 5 steps, Emden-Meyerhof pathway, which is, I want to say 12 or something like that. I don't remember the exact number. You know, you're going to have 30 or 40 steps that you could try to memorize. So to me, you're better off learning the rules, learning how to follow energy storage, whether things are anabolic or catabolic, knowing the definitions of those words, and applying them to problems than trying to memorize, you know, 30 different steps. And the other thing is there's a lot of other biological pathways that I can choose from, and that we won't cover in class, but I would expect you to be able to interpret. So I could even throw on a question on the test if I wanted to from the citric acid cycle, for instance. And how many steps are in the citric acid cycle? I don't even ever teach that one, so I don't know how many steps there are. It's like 6 or 8 or something, right? 10. So now we're up to 40 different reactions that you could try to remember. So I want to stress, don't memorize every process. Learn how to apply the rules and the definitions and steps. So here we've got energy storage. We've got reduced carbons up here because we reduced it. And then, of course, the other half of that process is going back to water and carbon dioxide. Whoops. Oh, good, it didn't delete. And, of course, we get energy out. By going through that opposite process. And so, for instance, this could be digestion. It could be the Emden-Meyerhof pathway, or it could be the citric acid cycle that we're talking about, since we're talking about carbohydrates. And is this going to be then oxidation or reduction? Oxidation, and this, of course, makes it catabolic. And so sometimes you can start to line up those things. You know, if something's an oxidation, you pretty much are almost guaranteed that the overall process is catabolic and things like that. And this is one way to picture all of these reactions. This is how I kind of like these pictures as good summaries of what's happening. If I were to write that as a chemical reaction, of course, you know, I'd have to write all the nitty-gritty details in there, right? We'd write CO2 plus H2O plus energy, and that goes to C6H12O6. Jay will be really lazy and write it that way, plus some O2. And then if we did another reaction, we'd reduce that back to CO2 plus H2O plus energy. So we're storing the energy, and then we're extracting the energy in those processes. So we can write chemical reactions like this, and sometimes we'll write chemical reactions. Sometimes we'll write both and sort of have the whole thing written like that just to keep it kind of honest. And I guess actually we could even point out that the energy is ATP. I have this as a random note written in my notes. I don't know if it's so miraculous anymore now that I think about it. I do think it's kind of interesting that ATP has almost been settled on by every living organism as the way to do things. But a certain part of me says that's kind of obvious. It makes a lot more sense that you would have one molecule that's good at storing energy that everyone uses as opposed to everyone solving their own problem, right? And if we really think about it in terms of evolution, if we really believe that everything kind of turned into or evolved into something more complex, then what we'd really realize is that it only has to be invented once and then everyone uses the wheel. The other way I used to think about it is in terms of money, because they always talk about ATP as being the currency of energy, right? Well, that's not a great analogy because how many different types of money do we have? A lot, right? And so I think that analogy is very poor unless you want to say that ATP is like the dollar bill and that it's accepted everywhere, basically, and that there might be some processes that use different energy molecules in different extreme circumstances, but pretty much ATP is it as far as the most generalized one. There are, of course, other examples of other molecules like GTP that we can use. We'll talk about, like I said, things like substrate level phosphorylation and things like that. Let's see. So there are 3 redox coenzymes that we want to talk about. Enzymes. And what these are is well, first off, what does coenzyme mean? They're needed in order for the enzyme to be fully functional, right? They're a part that goes with it. And what these are usually used for is the temporary storage of energy and transfer. One of the things that you're going to see is that we're going to run across these molecules in chemical reactions an awful lot. And if you can learn to recognize these molecules, which is the reduced form and which is the oxidized form, then this is one way to trace the energy flow in a chemical reaction. Now, Todd must go through some of these in Bio 204 because you do tests for them and various things like that. Do you guys know what these are ahead of time or not? In 111? I know since you do the citric acid cycle that you must have come across some of these at some point, right? So NAD plus, you remember those? What are the other two? FAD, no plus, NADP plus. And really, I probably should have written them in a different order. It doesn't really matter what order we cover them in. You do not have to memorize their structure. There's a reason that when we write them down, we write NAD plus instead of drawing their structure, because we would spend more time drawing their damn structure in a reaction than would be worth bothering with. What I do want you to know is have a good feel for them. Be able to, if you were given a picture, point out all the functional features of them, point out the part that's important in those molecules. And so, you know, you would be given the structure on the test, or you'd be given the structure on the cheat sheet. I can't even remember. I don't remember if I give you the structures on the cheat sheet or if they're even necessary. Maybe it depends on the test I do. So NAD stands for nicotinamide, adenine dinucleotide. So let's take a look at that. What are parts of that that you recognize? Yeah, adenine, you should recognize. We'll talk about what nicotinamide is. It's one of those things that pops up in 2 different places. And then dinucleotide, what is that kind of telling you is in there? 2 nucleotides, right? And so if we take a look at it, it really is basically just adenine plus ribose times 2 plus phosphate times 2 plus nicotinamide. And actually, I'm just going to save the FAD and NAD plus for another page. There's no way I'm going to try to cram it all in there. So if we take a look at it, the most important part of that molecule is what we call the nicotinamide ring. That's where the reaction occurs. All of the rest of it is stuff that doesn't matter, meaning it doesn't change when you look at chemical reactions. Whoops, that double bond is down here, here, here. C double bond O dash NH2. That's what we call a nicotinamide ring. And then what it really is attached to is a ribose, and then the ribose is attached to a PO4, is attached to a PO4, is attached to another ribose, and that's attached to my adenine. And so we don't care about those parts. Now, here's the key. I say we don't care about them, but I expect you to recognize them. But since you're given ribose on the cheat sheet, you're given adenine on the cheat sheet, and you know what PO4s look like, you should be able to recognize the full molecule. You can also look on page 942 in your book if you really want for the structure. And so what happens in this molecule is that we take a nicotinamide ring plus 2 electrons plus a hydrogen.
I don't think that is right. There's a baby sleeping.
924 isn't, oh, 924. I did the little transposition, sorry. That's okay. I'm like, I hear you go, there's a little baby sleeping on it. I'm like, did I draw something weird, or did I miss the baby sleeping in the room? You had me going there for uno momento. Maybe I'm thinking about babies because my poor friend was sick and her kids got like 104 fever. I feel sorry for little kids that get sick like that. I mean, there's absolutely nothing that they can do other than be miserable. So notice that if I'm drawing this reaction this way, I'm adding energy, right? And notice that I'm going to gain a bond to hydrogen. So what am I really doing in this process? Oops, not oxidation, reduction, right? And so the part of the molecule that changes is just that nicotinamide ring. And so here's the rest of it. I'm not going to bother drawing it because, well, it's boring. And what we do is we break a double bond and we add the hydrogens across it. So here's the hydrogen that we gained. That's this guy over here. Because really there's already one hydrogen there from the molecule being itself. So really what we do is we break a double bond. Oops, break. And add a hydrogen. Now, that's the last time you'll ever see that chemical reaction written, except for potentially a test and or a homework. Instead, we always use the letters in the abbreviation. So we'll simply write it as NAD plus goes to NADH. Meaning we won't worry about lots and lots of other details for this. And so what we really need to know is that this is the oxidized form, or what we call the low energy form, and that this is the reduced form, and this is our high energy form. Or if we take a look at it and we have a double set of arrows on it, then we can say for the top set of arrows that this is energy storage, and in this one we use energy. And so we can tell whether a molecule or whether a reaction gains or loses energy by simply knowing NAD plus and NADH. And that's one of those 50-50 things. You've got to figure out some way in your brain to make it obvious which one is the reduced form and which one is the oxidized form. To me, I simply say gain bonds to hydrogen means it's reduced, right? To me, that kind of makes sense. But I forget that I also do this all the time. And so you guys have to come up with your own rules. I can tell you my little rules like that to help me remember it, but at some point you really just have to make that effort. A certain part of me says that this chapter 33, 34, and 35 test, there's a lot of questions that are 50-50, meaning you can simply guess the answer. The problem is I ask so many of the questions that you better be guessing right, meaning you better know what you're doing. Because, yeah, you might get one 50-50 question right, but if I ask 10 50-50 questions right and you really are guessing, you'll only get 50% overall, right? Whereas if it was just one 50-50 question, you know, you might get 100%, you might get 0%. And so this last test on this stuff, if you really know it, it's kind of like the chemical reactions test, meaning it's not too bad in that you're just redoing the same bunch of rules over and over and over again. It's very easy to get a very high score. Conversely, if you really are only guessing at these, it's very, very easy to get a very low score. So this is one of those tests where you have to kind of know that material pretty well. It's kind of a hit or miss type test. We can do the same thing for NADP, except we're not going to show the details. Really, it's the same as NAD plus, except that there's 3 phosphates.
That's the one that's used in photosynthesis, right?
Yes. And then the other one that's interesting, because it's at least different, is that FAD. That stands for flavin adenine dinucleotide. And so notice that that's pretty much like NAD+. The only thing that's really different is the sort of molecule that we stick on it, this flavin versus, or the flavin rings versus the nicotinamide ring. And so this is made up of adenine plus ribose plus 2 phosphates plus flavin. I always feel like I'm cooking. It's like, I'm going to add some flavin to that. Okay, maybe that's just me. Never mind. I just ducked my head. I should also point out, maybe, since we haven't really done the lectures on nutrition, we should point out, like when we were talking about the NAD+, this comes from the vitamin niacin. And so that's why, for instance, it's considered one of the essential vitamins to have. Same thing with FAD. That actually comes from riboflavin, which is what we call vitamin B2. So these are important vitamins to have in your diet because they're used in a bunch of molecules that we like, or we use a lot. I don't know if we like them or not. Flavin. I don't know if I even want to do that one. Oh, did I already do this page? Gosh, Jay, good job. You're way ahead of life. So if we take a look at it, the flavin ring is actually 3 6-membered rings stuck together. And we've got nitrogens, methyl group, a methyl group, a nitrogen, bunch of double bonds, some more double bonds, some double bonds to O, an NH, another set of double bonds to O, a double bond. And then off of this middle nitrogen, we've got a CH2, CH, 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 CH2O, and then we finally go to our phosphates. And then I'm just going to, there, rest of the molecule. And then there's a bunch of OHs here, like that. And so that's what we call our flavin ring. And the parts of the molecule that are going to change, so if I add 2 electrons plus 2 hydrogen pluses, then I'm going to have the 6-membered rings again. Got my nitrogens. I'm just going to put an R here for the rest of the molecule because I don't really feel like redrawing the whole thing. And let's see, we still have our double bonds to oxygen, double bonds to oxygen. Again, please do not memorize this. You will want to shoot yourself in the head if you do that too much. What I want you to recognize is just kind of follow the reaction along, be able to make a comment about it if shown it, or be able to tell me whether it's being oxidized or reduced, things like that. I don't want you to memorize the shapes of these molecules. I mean, I don't think it's too outrageous to be able to go, oh, that's flavin, and then be able to recognize the nicotinamide ring. But I certainly don't expect you to draw it from scratch, like we would in some previous chapters with proteins and, disaccharids and things like that. And then the 2 new bonds, might as well put those in a different color just for fun, are here and here. So here is my oxidized form, low energy. Here's where I'm adding the energy. And I put that in parentheses because we're really adding electrons and hydrogen. It's not like it's energy when we talk about other reactions, but that's where it's being done. And this is our reduced form, or our energy storage. And so again, you have to be able to mostly recognize energy storage and energy, not storage. So if we were to write this in letters, what we would have written is simply FAD plus 2 electrons plus 2H plus goes to FADH2. And if we do our double arrows, that's a reduction reaction on top and an oxidation reaction on the bottom. So let's take an example of what would be maybe a test question or a homework question or something like that. So you'll, in general, be given the reactions. It might be possible that I would write a sentence where you would have to convert like the word ethanol to what that is, or ethanol, or something like that. But generally, I'm not trying to test your knowledge of organic naming, although like a homework might have a question like that. But on a test, I usually will give you the structure of the molecule just to make life easy. So if I have a process like this, 2FADH2 plus oxygen goes to 2FAD plus 2H2O. Questions that we'll ask, for instance, are, does the process gain or lose energy? What's being oxidized and what's being reduced? So, basically follow the energy. Talk about what's oxidized. Talk about what's reduced. And so, like, for instance, where should energy be added on this reaction? On the left side or the right side? Where should energy be added? Follow the energy, or conversely, where should energy be added? And I should also point out that I can also write this same thing and simply say, here's a reaction, add FAD and FADH to it, meaning the inverse of the problem, right? Maybe I'll give you a separate question on that. There's so many ways that you can ask these questions that you really have to just be able to know what's low energy and high energy forms of molecules. You have to understand oxidation and reduction, because there's no way you can memorize every possible example I can give you. So does energy go, for instance, on the right side or the left side? Yeah, so if we were kind of answering the question, we'll use a different colored pen, maybe. Hello. What's going on? What? Do not do this to me, computer. See, it's moving. I'm writing. Why won't it let me select a pen color? Why won't it go from one slide to another? This is very strange. Why is my pen broken? Why does... No, it's not a loose plug, Jay. Did we kill it? Apparently we did. You guys got that all right down? Yeah. Okay, so I'll kill it and I'll rewrite some of it up. I probably won't write all the darn questions up. I tell you, this thing works like for weeks in a row, and then all of a sudden it just says, you know what, Jay? F off. Oops, so if we write our reaction, 2FADH2 plus oxygen, 2FAD plus H2O. Where should we add energy in that reaction? Yeah, we agreed that it should be plus energy. Now here's the question. So you've got a 50-50 chance on the test to do that, right? Why did you choose that side? Because it's losing bonds to hydrogen, right? And so see, that part is because you recognize that this is being oxidized, right? Because we lose bonds to hydrogen. Or you can recognize that this is the high energy form and this is low energy. And so to get from high energy to low energy, the energy has to be the same on both sides, right? To within thermodynamic limits. Sometimes I feel like I say those things because I'm afraid someone will watch this lecture and go, man, he didn't even mention that you always lose some energy as waste heat. And then I'm like, man, shit, no one's going to listen to these anyway, so maybe I shouldn't even open my mouth. But I do anyway. So high energy, low energy, so obviously we need to add energy to the right side of the equation to make that happen. So if that's being oxidized, what's being reduced? Because it's gaining bonds to hydrogen. So if we take a look at it, and see, this is one of those where it's a little weird to think about sometimes. What's the oxidation number for just an oxidant, oxygen? Because usually when we're following these reactions, 9 times out of 10, it's carbon. It's 0. Because overall oxygen is neutral, right? So each one has to be 0. Whereas over here, that oxidation number is +1, -2. I'm talking about oxygen. And so we can see that the oxygen is reduced. It gains bonds to hydrogen. Its oxidation number is decreased. Because remember, negative oxidation numbers are good. They mean something's reduced. Actually, it's not like they're good. I guess they're not good and bad. They're not evil or anything like that. So this is actually a step that we don't really talk about very much, but this is how we transport electrons to mitochondria. Mitochondria. Or the electron transport system. Yeah. Essentially. So what we're going to see, now remember we said that like things like FAD, H2 are responsible for the transport of energy. So they're transporting energy from some chemical process and then releasing those electrons to be used in the electron transport chain. Oh, that's interesting, but we won't worry about it. About 85% of the energy in a cell is actually used directly, meaning it's simply extracted and used immediately. Only about 15% is stored in ATP to be shipped around. And we've already looked at how ATP stores energy, right? That was way back in chapter 31, which really wasn't actually that long ago. But how does it store energy? Just, what word should we be associating with this? High energy phosphate and hydride bonds. There is a section on your book talking about molecular oxygen and the formation of hydrogen peroxide and the formation of molecules like superoxides and stuff and why it's bad. And it's kind of an interesting read, but it sort of is, I consider it an offshoot. So like for instance, if you're studying, just go ahead and skip 33.4. We won't have questions based on that. Oh, look, there's ATP. What do you know? I don't even know why I have it drawn in there again. Let's put it this way. If it freaking shows up on the test, say, Jay, go look at lecture from today where you said there's not going to be anything from section 33.4, and I'll take the points off, and I'll give you 50 points extra credit. So no, I'm not going to slip up and accidentally put a question on there on this section, especially if I remember explicitly stating that I'm not going to. So admittedly, I have to be honest, in some chapters I told you I'm going to skip stuff and it might show up on the test as a couple points on a question, right? Here I'm telling you the opposite, that I'm going to skip stuff and not going to show up as points. Okay, so let's see. These are two big topics. I'm trying to decide if it's worth trying to do both of them. Maybe we'll just do one of them and then you'll end up having to do the other one. You won't be able to quite finish the homework, I don't think, but you'll be able to do the other one. We can finish the other one later. So if we take and talk about how we can convert energy. So this is energy conversion. And what it really is it's the method to convert between our two normal storage forms of energy. Method to convert energy between storage forms. And there's a number of different ways to draw these diagrams. We can look at the actual chemical reactions, but I've also told you that I kind of like looking at those energy level diagrams they have. And so what we're saying is that if we have an energy level diagram, that we have as high energy reduced carbon, and that if I oxidize that, so here's my oxidized carbon, and notice I'm being a little vague because we're just trying to show it. So this is plus O2, this gives plus CO2 and water. The idea is that we take that energy that's released and we need to be able to convert it into a different storage form. And so the way we do that is we can take substrate molecules, and this is essentially, and I'm going to put this in quotes, any carbon containing compound. But what we'll find is that there's some that are more used than others. And we make high energy phosphate bonds. So this is one method. It's called substrate level phosphorylation. Oops. Phosphorylation. And there's two methods I should point out. So this is the first one we'll talk about. And so this is a way that we can convert energy from being stored in carbon and oxygen bonds to being stored in phosphate bonds. And then once those energy bonds are stored in phosphates, then they can be transferred to an ATP molecule. And so that's a pictorial diagram up above following the energy. If I were to write that as a chemical reaction, what we usually take is some molecule that has an OH group on it. And here's where you have to use your imagination just a little bit, in that I can draw it, and maybe I will draw an example next, where it's a very specific molecule. But what you have to do is say, okay, when I see this process occurring, even though the molecule itself can be different, because remember, R can stand for anything, right? That I'll kind of pick up on that, plus a phosphate, P-O, double bond O. And whether that one is shown again as an O- or an OH is book dependent. I'm going to follow what your book does and show it as an O-, plus energy. And this is from a redox reaction. Then I can, of course, minus H2O reaction, because, well, 90% of reactions in organic or in biochemistry are minus or plus H2O, dash O, dash P, dash O-, double bond O, dash O-, plus H2O. So what I've done here is I've made a high energy phosphate bond. Between the, oops, what were you going to ask? Sorry, I thought you were going to ask about where the arrow is pointing, because I did notice it points to the wrong spot. High energy phosphate bond between the substrate and the PO4 group. What were you going to ask, Brittany?
So it's the bond between
Oops.
the R or the rest of the phosphate?
This one right here. Your book's nice in that it actually draws it as a different color. For instance, we're going to see this reaction in the Emden-Meyerhof pathway. By the way, I start using a lot of abbreviations. You should recognize that EMP after a while stands for Emden-Meyerhof pathway, because quite honestly, it's annoying to have to spell it 50 million times. And so then what happens in the next step, is we can take that R-O-P-O- O bond O, OH, plus an ADP, we can get an ATP plus our OH. And so notice that our substrate can be reused. Or maybe you want to say recycled. That's one of the things you'll notice about biological pathways. I'm sure Todd's pointed this out, that there are molecules that link the pathways, and that usually the pathways recycle all of the reaction molecules that you need, or at least all of the energy molecules also that you need.
So is that why there's such good energy storage that's
I don't know if I would say that this is such good energy storage. It's simply a mechanism by which we can transfer a phosphate from a substrate to an ATP. And so it's an intermediate step in the transfer of energy. I don't know if there's a specific reason why this step is chosen among others. One of the primarily reasons that this is used is it does not require O2. So that means it's anaerobic. That's one of the big advantages of substrate level phosphorylation. And I also mentioned it's already used in glycolysis or the Embden-Meyerhof pathway, used under anaerobic conditions. And this is really the only source of ATP under anaerobic conditions. And it's basically only for small amounts of energy. And it's very inefficient. Maybe I'll give that its own bullet point. What the hell? Very inefficient. So by choice, we don't use this mechanism. But where might it be used? Or when might it be used? Right. What conditions would have no oxygen? Because usually without oxygen, we're dead. Just so you know. Well, this would be used like in your muscles once you've kind of gotten rid of your normal ATP storage, and if you don't have enough oxygen getting to them, this is the only method by which you can do it. There are also certainly you guys talked about anaerobic versus aerobic bacteria in class, right? In micro. So I mean, there certainly are places where there's anaerobic conditions. For instance, like the bottom of lakes and stuff, you see a lot of anaerobic processes that occur. They usually occur very slowly because this is a very low energy type of process and it's very inefficient compared to other forms. It's more of a fallback mechanism if there's no oxygen around. If there's oxygen around, we don't tend to use this very much at all. Okay, so if we wanted to draw a specific molecule in there, I guess we'll draw one more example. So I guess this isn't a reaction. This is just an example, I guess. I don't know. I wasn't going to show. I guess maybe I'll show it. So if we have this molecule, and we react it with P double bond O, O minus, OH, HO. It can, of course, react with either end of the molecule. It doesn't really matter which one we show it reacting with. We can show it reacting with the middle, and then we get C double bond O dash OH dash C. Oops. I guess if I'm being really good, I'll put the CH. Oh, no, never mind. There's no need. There's a double bond CH2 dash O dash P dash OH double bond O, O minus. So here's our high energy phosphate anhydride bond, or HEPAB, high energy phosphate anhydride bond. Sounds good to me. I'm too lazy to write it. I've never actually used that as an abbreviation. It kind of comes out nice. And technically this molecule is I don't expect you to memorize this, phosphoenol pyruvate, and it's from glucose metabolism. Meaning you will see this again when we talk about the glucose metabolism in the product. Oops. Okay, technically that's the molecule because it has the PO4 on it. Oops. That would just be pyruvate on the left-hand side. Anhydride bond. Some books will just say high energy phosphate bond. I like to throw the word anhydride in there. It's one of those, again, 50-50 things I've seen is whether they show that as O minus or OH too. One of the things that bothered me when I first started teaching this class, especially because, like, if I write a test, I go on the internet and I just search for various molecules to use in chemical reactions instead of drawing them myself because I'm lazy. And so it's like half the molecules you're like, oh no, can't use that as an example, can't use that. And so like my first few tests, I didn't pay attention to things like that because it was just made sense to me. and confuse students. So I try to look for the molecules, and I'll try to find pictures that draw them this way. But you know, if occasionally it has an extra OH on there, don't freak out. Is that fair enough? Okay, how much time do we have left? I guess we're kind of over already, right? But that's okay, we're getting Friday off. I'm gonna finish off one thing just because I think it allows you to finish off the homework and it'll kind of finish this off. The second one that we can talk about is oxidative phosphorylation. And of course, this uses energy directly from the redox coenzymes, directly coenzymes. So that's our NAD, NADP plus FAD type molecules. This requires oxygen, so it's aerobic. It's very efficient and can transfer large amounts of energy. We'll use it when we talk about beta oxidation and the citric acid cycle. So this is in chapter 35 and chapter 34. See, again, we're talking about oxidative phosphorylation now. We're defining it. And then when we get to the chapter 34 and 35, we expect you to say, oh, that step is oxidative phosphorylation. And things like that. Meaning this chapter is all about recognizing everything and doing that. And it's also used, or it occurs, in mitochondria. And so maybe I should make a point on substrate level phosphorylation basically occurs anywhere, which really means anywhere but the mitochondria. Just on under phosphorylative substrate level phosphorylation, sorry, let me get that straight. It occurs, if I remember right, it's the cytoplasm actually, but I might be wrong on that part. Basically, it doesn't occur in the mitochondria is what I'm trying to say. And so here, if we kind of draw it, this is really just in your book. It's figure, one of my favorite ones in your book, figure 33.8. I'll even get the page number right, 930, no transposition of stuff. And there's two of them that we're going to worry about. Basically, we can have FADH2 plus a half O2, FAD plus a half O2. So this is mitochondria. And then the energy is transferred into something useful by taking 2 ADP plus 2 inorganic phosphates and up moving them to 2 ATP plus 2 H2O. And I guess, I don't know where you draw the lines. So essentially this is if I wrote the reaction route, FADH2 plus 2 ADP plus some oxygen plus some phosphate goes to FAD plus a half O2 plus 2 ATP plus H2O. And so what we're really saying is that one FAD H2 is essentially equal to 2 ATP. Oops, that's a 2 ATP when we do the math. So you guys have already practiced this. I know, at least, I don't know, maybe Todd didn't really do it in class, but he certainly, the extra credit assignment people brought around to me, you had to know and, you know, convert FADs into ATPs and things like that. Now here's a tough one. well, I won't say a tough one. This is just another one of those 50-50 things. Obviously, I'm going to write the reaction next for NAD and NADH and NAD plus and all that. How many ATPs is that worth? Well, an FADH2 is the equivalent of 2 ATP. Do you know what an NAD plus or an NADH is worth? 3. So that's another one of those 50-50 things, right? One's worth 2, one's worth 3. I can never remember which. That's how I remember that it's 2. And I don't know, maybe you guys are really good at memorizing random facts like that. I always have to have a trick for 50-50 things. I remember my first year teaching, I had 2 girls in class, and I constantly swapped their names because I don't know why. I did. It was my first year teaching. It was horrible trying to remember anyone's name for that matter. Probably the worst part about teaching was trying to remember names. And I finally came up with some stupid thing, and I don't even remember what it is anymore. Like one of them might have had curly hair, so I said, curly hair is Karen. Something silly like that. They both kind of start with K, even though curly starts with C, but you know what I mean. And so that was the only way I could remember them. So the other set of reactions, so what goes on top, NADH or NAD+? What's the high energy form? See, here's where that's like a question that you kind of have to know. Yeah, NADH is the storage form, so that means it has high energy, plus H plus a half O2. Don't memorize these reactions, only know the conversion ratios essentially. Now, do memorize or do know which forms are high energy and which forms are low energy. Meaning, I'm not going to ask you to memorize a whole list of chemical reactions. I'm going to ask you to memorize a few simple facts so that we can follow the energy level or the energy transfer around there. So here's my energy being transferred. We said that in this case it's 3 ADP plus we of course need 3 inorganic phosphates to go to 3 ATP plus 3 H2Os. I don't know why I draw lines. They draw lines in their diagram. Actually, they use boxes. And so here, if I was writing out the reaction, we could write NADH plus H plus a half O2 plus 3 ADP plus 3 PI goes to NAD plus H2O. What's what? What does what mean? That's a D. This is a D, right? Oh. Okay, no, that was a P. It was supposed to be a D, though. See, if I'm saying ADP and I'm not writing D, when I say D, then I might write an extra P. Good catch, though. It means you guys are paying attention. I like that. What am I doing? Was that H2O plus 3 ATP plus H2O? And yeah, I could add the H2Os together and make 4 of them, but I'm just kind of doing that. So, like for instance, a test question might be, and this is a good way to point out like how I test about things, since it's been tough lately. I might write this. And you have to remember where the ADP and the ATP go. Or I might have an energy level diagram and have you fill in the boxes. The idea is that I want you to know which forms of molecules are energy storage and energy transfer and be able to pair them up properly. And so really you need to know which forms are reduced, which forms are oxidized, which forms store energy, which forms are high energy, low energy. There's a lot of different ways to say that in all of the same thing, kind of. So let's stop here. The rest of the week, Thursday is work on lectures, work on chapter 32 presentations. And then on Monday, Friday is no class. And again, Thursday, if you want to come in and ask questions, work on stuff, make sure things work with the computer, things like that, good time to do it. If not, you know, Monday is sort of the last chance to give me stuff to copy. Let's say homework 33A is due. I forgot there was a 32C even, so that really should be due today too. But go ahead and turn it in tomorrow or Monday. I don't really care. I mean, we're not having the test till Thursday, as long as you get it into me kind of by Monday. But yeah, you're right. I did forget about 32C. So we'll say 32C. It's, you know, it really should have been due today since we finished on it. But if you turn it in Thursday or Monday, I'm not going to be terribly disappointed. Tuesday, we're going to do our presentations. And we'll do them in the order that they come up in the book, so whatever order that is for people. Wednesday, we'll do chapter 33. And Thursday, plan on a test on 31 and 32, of which you'll know all the questions, or at least 90% of the questions for chapter 32. So really, you're only having to study chapter 31 and the parts of 32 that other people did, kind of. So I hear you guys get to start on your uber unknowns for micro. I always think that's the funnest part of that class.